It could go long. So this afternoon, I want to welcome Dr. Horacio Lopez-Nicora from The Ohio State University. He's currently an assistant professor, soon to be associate professor at Ohio State, where he works in soybean pathology, nematology there at Ohio State. He's originally from Paraguay. And got a degree in agronomy at the National University of Ascension. That's how I'm going to say it in English, right?
Sounds great.
And then from there, he got actually a Fulbright for his master's degree to work in plant pathology at the University of Illinois with Terry Niebleck. And from there, followed her and worked on his PhD in plant pathology at the Ohio State University again, looking at SCN, soybean cyst nematode, and macrofemina interactions. In addition to getting a PhD in plant pathology, he also got a master's in applied statistics. So Horacio is someone we rely on when we do some of these national group kind of stuff, to do some of the stats and things. And then after that, his PhD, he went back and spent 3 more years in Paraguay, and then has come back to Ohio State, where I've gotten to know him starting here. He's a colleague of mine. So we send him a lot of soybean cyst soil samples and things like that to work with. So welcome, Horacio, and I'll let you.
For her.
Yeah,
That was a great introduction. Yeah, thank you, Darcy. That was
welcoming you.
Thank you, guys. It was great to visit with most of you today. And thank you, Darcy, for the invitation. I think it is recording.
Yes.
Yeah. Okay. Cool. So I'm gonna go ahead and set a timer here, or else I'm not gonna shut up when I talk about these organisms. It's gonna go off 3 times, and that will indicate that the 3rd time, and I need to start backing up a little bit. But I do work with many diseases that affect soybean. Pretty much anything that affects soybean falls under my empire at The Ohio State. But soybean cyst nematode is one of the most important one, and we'll talk a little bit about why. And I'm gonna show you a little bit of things that I've been doing as part of the research when I started at OSU since 2021, and some of the ideas that I've been developing, and hopefully I'll get some good comments and feedback from you guys. So let's see if this works. Okay, cool. So to keep myself on track, I prepared this agenda. First, I want to talk about why soybean cyst nematode is the most economically damaging pathogen of soybean in North America. There's absolutely no doubt and reason for us to argue that. And I'm gonna try to explain why. It's an evolving pathogen, and how we try to quantify that evolution in Ohio will be the most natural way of talking about soybean cyst nematode, and then integrated management, how we can combine different things that we can do to help growers, first of all, stop or reduce the shift in virulence, but at the same time protect yield. And then last, I would like to talk a little bit about future and impact that we are doing by gathering all the information we have, preparing communication materials and resources, and then sharing it with growers and anyone interested in learning more about this pathogen. I am the co-leader of the SCN Coalition, the scncoalition.com. You'll find a lot about soybean cyst nematode, and that will be the portion that I'm gonna be talking about there. So, good plan, right? Yes. There's room here for anyone who wants to come. I do speak a lot when I talk. That's because I'm excited. And the first thing that as plant pathology that I use to try to explain how damaging this pathogen is using the life cycle or disease cycle. It's more appropriate to call it life cycle here. It's an animal like you and me. And it will overwinter as eggs. And you can see that some of those juveniles are coiled inside those eggs. They will molt for the first time within that egg and then hatch in this 2nd stage juvenile, which is the only infective stage of all the life cycle of the nematode. And they will be able to infect the root using that stylet. It looks like a little dagger that they will use to penetrate the roots. These are roots that we stain with a stain that will stain animal tissue, because then we can track the nematode and see what they do. So we can see that the nematode will migrate to the vascular cylinder and start interacting with cells. And they will become sedentary, stop moving for the rest of their life. And those cells will change anatomically. If we cut those cells very thinly, we will see that the anatomy of cells that are in the root system in this part transform And we see patterns or anatomical features such as transfer cells that we know exist in the embryo or in some part of trafficking nutrients. So the nematode is taking control of the plant machinery to make those plants feed the nematode. They will continue growing. The males will regain this vermiform condition and they will excite the root to localize a female to fertilize them. So this is an obligate sexual animal, just like we are. The female will lay on average 250 eggs. And this can happen 3 to 6 times in one season. So conversation with growers saying, I have little soybean cyst nematode I don't have to worry about. It's very dangerous because that starting with very little nematode at the end of the season can really end up with above damage threshold because of the way that the nematode reproduce so fastly. Now, how do we identify that we have soybean cyst nematode? The root will not lie. If we pull those roots, we will see those females attached to the root, lemon-shaped, whitish at the beginning. When they die, they turn brown, very, very tiny compared to nodules. We want a lot of nodules on legumes because they fit atmospheric nitrogen. We don't want the little female cyst attached to those roots. Now, the symptomology, so this is the sign, right? Plant pathology 101, the sign manifestation of the pathogen. The symptomology of the disease will show up more severe when we have abiotic stressor. So if we have, for example, nutrient deficiency, let's say potassium, marginal chlorosis in the presence of the nematode, more of that marginal chlorosis. SDS pathogen causing SDS, sudden death syndrome of soybean. In the presence of the nematode, more SDS. Resistance versus susceptible in sandy soil. Susceptible, very stunted. Root, very stunted in the presence of the nematode. However, this is an exotic pest that evolved very well with a very good exotic host that we brought and breed and developed in the United States. And this is the nightmare situation that we live in the north central region. Plants that look extremely healthy, yet when we go and harvest that susceptible cultivar and compare it to the resistant one, we can detect significant yield reduction with absolutely no visible symptoms. I had the best soybean ever, and you drop and left behind 30%, 37% of your yield. That's a lot of money. And every time I say that, I question myself, is that true? I mean, is that possible? So in 2021, when I started, I put this experiment in a field infested with nematode, 100 grids that we went and sampled each one of these grids and collected soybean cyst nematode, initial population. That relates pretty well with disease yield reduction. And you can see the pattern, right? This is the type of beast that we work with when we're dealing with soil-borne pathogen. They're aggregated in space. You have very red quadrants that indicates a lot of nematodes, above 1,000, 5,000 eggs per 100 cc of soil, cubic centimeters, and blue quadrants that we did not detect the nematode. So no detection of the nematode. We collected yield and then we went and we measured everything to try and capture difference in symptomology, height of the plant, branching, numbers of everything we could, intensity of the color of the foliage, no visible symptoms across that field. Very homogeneous growth of soybean. Now, during my PhD, one of the things that I started thinking a lot about was how many statistical models assumptions we violate by assuming that this data is generated randomly, right? We do a regression, which is something that we may do to estimate how much yield reduction we see here compared to here. And we assume a normal distribution, identical and independent distribution with mean of your residual zeros and what else? Variance equals to variance. So the variance are assumed to be the same. The covariance are 0, no correlation there. And the observation is independent. And then as students, as researchers, we go and we freak out about normality all the time. Normality, we just want to see our QQ plot. Normality truly can be severely violated and our models are very robust. Heterogeneity in the variance and the lack of independence in your response can destroy your model's inference, which means that you can be calling significant to something that is not, or you can miss to call significance between treatment when you don't account for the spatial variation. So there is, I hope you can see a spatial aggregation. So what can we do? And I will refrain to put mathematical equations. If you do want that, go to the different reference that I will provide you. But there are techniques that we can use to account for that. One of these is using this type of contiguity matrix. So what we're saying is we are assuming that the pathogen, has this spatial autocorrelation in a way that is diagonal, and we use the Bishop contiguity matrix. And it's lovely, right? Because we love chess, right? And then, well, no, actually, the aggregation goes like this, vertical and horizontally, row, column. But that may not be naturally how it occurs. And then the queen is the one that can move everywhere. And we're assuming that that's the way that maybe the pathogen is moving, because we know teal, et cetera. You make a lot of biological assumptions too. But by using this, you're accounting for the spatial aggregation. You're taking that into consideration. And now the observations become independent. And you can run your ANOVA with the assumptions checked. Does that make sense? That's what we want to do, right? And we can check this with residuals, et cetera. And that's what we did in this case, because we wanted to ask the question. And again, sometimes I get hung on the model, and it's fun, right, doing math. But we need to think about, okay, what does this mean, right? At the end, I want to know how much nematode I'm losing when I'm not even seeing any symptoms. Box plot, very simple box plot, a lot of information here. The wider your box for those red quadrant, high numbers of nematodes, the more variability. Of course, we have levels of 1,000 to 5,000. The tighter your block where we did not detect nematodes, the tighter your box, less variability. That's soybean without the pathogen. Yield on your y-axis, average on those lines. Simple math, we're losing 37% with absolutely no visible symptoms. Matched. Whatever research have been showing us since 2003, Wang et al. 2003, holds in 2021. At the price of soybean, I hope we get there again sometime, $300 an acre. I mean, it's huge. You could take your family to Disney, buy yourself nice shoes. It's a lot of money. We don't see it. We think that the soybean grew very well. This is the reason I think this pathogen remains the most economically damaging pathogen of soybean. SDS will tell you it's there. YMO will tell you it's there. You can smell it. You don't see it. You think that your soybeans are very healthy, yet you're losing.
The growers buy these stories, and the growers, they don't see it, right?
Yeah, exactly.
And
exactly. The researcher don't see. The researcher goes and measures and cannot capture that.
Yeah.
Unless you go and harvest those areas where you sample and you identify the hotspots. Yeah, so what do we do? Great question. So sample your field, it's easy one. And we all, as agronomists, know how to do it. Whatever we're gonna go and start a crop, we take a soil sample for soil analysis. Take a subsample of that and send it to the nematology lab. You will get information about your nematode community. You can dig plants. I don't like this one because you sometimes just don't see it. Or you pull that plant that really doesn't have the nematode, 3 feet from there, tons of nematode. You just miss it. A soil sample, composite soil sample is better. Good. Classical way of soil sampling, what I recommend normally to growers is go to the spots where you're yielding low. Why have you been yielding so low? You don't even know and explain why. You have addressed many things such as nutrient deficiency, yet your yield map is showing low yield. Maybe that's soybean cyst nematode there. And these are all some of the resources that we generate in the SCN coalition, right? And I'm gonna talk more about that. What do you do with your numbers? Okay, you sample, you get your numbers, you report those numbers to the growers, know your numbers, how much soybean cyst nematode you have, what do you do with that? We normally provide a table saying low, moderate, and high. What I call it moderate, Indiana may call it high. What I call it high, Iowa may call it low. And we're confusing consultants, right? So the SCN profit checker calculator, also developed through the SCN coalition, builds up on top of 35,000 research plots. that is running behind an algorithm of a stepwise regression that is taking this input. How much nematode you have? What's the percentage of sun, texture? What's your soil pH? And then what's the SCN female index on PI 88788? This is the first time I'm introducing PI 88788. I'm gonna probably say it 75 times today. But PI 88788 is the most common source of resistance used for soybean cyst nematode. As a matter of fact, it's in more than 90% of the commercially available soybean. As a matter of fact, I cannot find susceptible soybean anymore that doesn't have PI 88788. As a matter of fact, we will all as growers, forced to use 88788 without being forced really, because the market only had PI 88788 wherever we planted soybean. Okay? And the female index is how much that population can reproduce on 88788 compared to a susceptible. 33 means 33% compared to a susceptible. Yeah?
So that's not fully resistant, it looks like.
In that case, no. But we will see that some are more and or less. Yeah, quantitative resistant.
Can you explain how a grower gets that number?
What's that?
To get the female number, there's no way to do that.
So this one here, the female index is required for this calculator to run. It costs, in my lab, $100, and it takes at least 2 or 3 months to get it. We have to increase the population from your field. Then we have to put it in differentials. I'm going to show that in detail. And then we report that, right? That's the first one. We provided our average from the state. So if you're in Indiana, you have provided that average female index on 88 and the grower can go and choose Indiana and use that. Pretty good. It's the average. Works fine.
What is
Better
it?
than nothing.
40?
40, yeah. Yeah, ours is a little bit higher too. This is from Nebraska, I think.
I see.
So what do? We with Ohio Soybean Council supporting this, we receive samples from growers and we run those samples and report the numbers of soybean cyst nematode to them. And we have been doing for a long time. So I started in 2021. We have been processing more than 400, last year, more than 500 samples to growers. And that allows us to create this type of map where we know where soybean cyst nematode is and how much on average, right? It also allows us to track counties where we're receiving samples and they're normally non-detected. Oh, now it showed up. Places where we didn't see soybean cyst nematode, that now we're seeing soybean cyst nematode, expansion of soybean cyst nematode. So far, out of 2,200 samples that we have processed, more than 60% have soybean cyst nematode. The next person from Ohio that comes through the door, I will say, hey, you have soybean cyst nematode for sure. Which is a bad news, right? And my mom always say, if you're gonna say something bad, follow up with something nice. Fortunately, in Ohio, most of these samples are low to trace, which is great because one of the recommendations to grower is don't stop growing soybean. Just try to keep those numbers low because it's hard to bring high levels to below damaged population. Okay. I know you're asking yourself, well, I mean, you have all these soybean cyst nematode populations. Are they all the same? No, they're not the same. They differ in the way that they reproduce on sources of resistance. And what does that mean? Well, if we go back to 1954, North Carolina reported for the first time, then soon after, soybean cyst nematode everywhere in the US, agronomists, breeders, plant pathologists, nematologists realized how much of an impact it could cause in the soybean producing regions. So they went out and tried to identify sources of resistance, and they found out on these germplasms or PI, plant introductions. And they find it in a chronological order, which is the numbering that the AG type takes. I'm gonna talk about AG type. But the king is one of them, PI 88788. This is the second time, so I have 73 more times. And then other ones all the way up to cloud. Now, think about this. We have all these sources of resistance that probably you have it on your breeding programs, but only 2 are available to growers, commercially speaking. Peking and PI 8878. This one here, number 4, PI 437654, is a super powerful resistant line. And we always include it in all of our testing, because if we identify a population that can reproduce on that one, we're lost. Now, they figure out that yeah, 88788 worked really well and was very easy to introgress in high-yielding soybean varieties. Soybeans that we like to grow, that we knew how to grow, and that continue yielding the way that we wanted them to yield. And we were able to put that trait that worked, as you can see, very well against that population, AG type 0, because it cannot reproduce on any of these 7 sources of resistance, compared to a susceptible. And that was great. Now, what we're gonna do, we're gonna put it in all of the soybeans that growers can have access, and we're gonna plant that same source of resistance over that same population for 30 years, non-stop. What happened? Huh?
It overcome, yeah.
Overcomes the breed.
You exerted a selection pressure and it shifted the virulence dramatically to one that can now very easily reproduce above 10% compared to a susceptible. That above 10% is technicality. The way that they come up with that, they figure out we have 10 fingers and they say 10%. No, to be honest, I think they borrow it from the SMAT rating, and that's the way they use it. There's no explanation really. But the female index can become into a very quantitative data that we can use rather than calling things AG type 0, AG type 2, etc. Well, AG type 2 reproduces on PI 88, Now it makes sense that if it reproduces on Peking, we're going to call it AG type 1. And if the population can reproduce on PI 88 and Peking, we're going to call it AG type 1, 2. Very good. Okay, so I need water. So in Ohio, we went back 1996. There was a nice distribution of heterodera glycines in Ohio, and you see a bunch of massive amount of sampling that they were processed, seeing that the nematode was really not present in many places. You see a lot of holes, out there. The triangle, the black triangle, those are the ones that tested positive for SCN. So a lot of sampling, not too much SCN.
Nowadays, how does it look like, the sampling in Ohio?
¿Cómo? What?
Is that similar to what it used to be like that, or
So
less?
20 years later, I say, well, let's do the same thing and now figure out what's going on. And in most of our sampling, more than 85%, we had the presence of the nematode. We tried to go to the same areas that the previous one showed to try and see if those places that didn't have the nematode now have the nematode, and they did. So look at the trend here, 1996, 2016. So that 6 will follow us a little bit. But 2 things that we wrote in this paper. One was that we were reporting also abundance, how much we had, not only presence, absence. And the other thing is that we couldn't, with the information we had, tell what virulence profile the population of Ohio was. Can it reproduce on Peking? By how much? On PI 88 and 88, by how much? And that's where Gilly comes in. Gilly is a PhD student in my lab, and he decided to grab those 2,000 populations. Well, actually, he didn't decide. We figured out that he was really good and fast at getting these phenotypes. He was like, when he came, no, I shouldn't. No, we'll talk later about Gilly. So he selected 150 population that he phenotyped in 1 year. This is super fast. And the way that he does it is he grows the inoculum, prepares the inoculum, and then he inoculates the 7 differential. You see this? The 7 differential compared to a susceptible, which is Lee, and generates this female index. Percentage compare relative to susceptible, right? Of course, we realized, why are we putting all these if commercially we don't have it? So we changed it to only use Peking, PI 88 and 88, and that number 4 that is super resistant. So that's what he did, and generated female index on Peking and PI 88 and 88 for 150 population. Now, one of the things that I was thinking, he was presenting this data, and I went back to this paper that I read so many times when we were writing about the distribution of soybean seeds nematode in 2016, I never, for all this year, look at the table that paper showed. And that table essentially is exactly what Geely did 30 years ago. With the standard deviation, with the mean female index on Peking, on PI 88788, and on other stuff that we don't care for now. Maybe in 10 years, I'll be like, what Pickett is there? So this is great because now we have the same data set with variation that we can use in an analysis to compare what really happened in 30 years of planting whatever soybean we have been planting in Ohio. So to do that, we use a Bayesian hierarchical model that the data itself is structured hierarchically. So the samples come from the county. The counties are the fields, and then you have variation when you go and collect that. And then Geely puts some more variation when he runs the female index, right? And then we model female index as continuous response. We did a logi transformation, but now we're moving away of AG type 0, AG type 1, 2, or whatever you want to call it. We're focusing on what's the percentage reproduction on Peking and PI 88788 compared to a susceptible. We now can estimate change over time, capture the spatial variation, because now we can see, well, for what counties we're seeing more adaptation or more shift in virulence. And then we also account for that uncertainty of our sample. So this is one of the graphs that we can see that 30 years ago, we see, for example, for PI 88788 that we increased 14% point. Wherever we were at that time, now we increase significantly high. And I know that the line is kind of redundant because between 2 points, the only thing you can have is, and the shorter distance, a line, right? It would be great to have more years here so we can see how that behaves, regardless. 5.2 for Peking. This is concerning, right? Because when you take the fold of this, the fold increase, we're seeing a 3-fold increase for PI 88788 and a 7-fold increase on Peking. And Peking should be the one that is not commonly used and should be the one that we want to use to protect our soybeans in places where PI 88788 lose effectiveness in resistance. This also shows exactly the spatial shift in virulence. If you can see here, all the dark dots are Peking, all the lighter dots are PI 88788. And really what I'm trying to show you here is that when you look 30 years ago, everything was clustered in a very small female index. They were not reproducing on our sources of resistance. 30 years later, not only those medium or mean shifted and now they can reproduce at very high levels, but the confidence interval shows significant variation. So you see in some fields, you have Peking allowing for almost 40% of the reproduction compared to a susceptible. Insane, right? And then a map shows this very nice. You can see that 30 years ago, the map was a little bit more blue than the one right now that is more red. I'm not talking about politics here, but the blues are the AG types that didn't reproduce on any source of resistance. The red is the one that can reproduce on 88788. The green, which is my NIMER, populations that can reproduce on both Peking and PI 88788. What are you going to do if you're a grower that have a population that can reproduce on both PI 88788 and Peking? The only 2 sources of resistance you have to manage the nematode. And this hopefully, takes the message home that the shift is happening, right? So to answer my question, sample your field, know if you have the nematode, rotate sources of resistance, if you can, rotate varieties. Even if you have 88788, by rotating varieties, you can help with stopping or decelerating the shift in virulence. Rotate to a non-host, we do this already very well here in the Midwest, so that's good. Planting corn or small grains will not allow the nematode to reproduce. And then consider a seed protectant. Consider a seed protectant. And we have that message there because we don't see consistent result in reducing nematode population. And I'll show you some of those results. So that'll take us to the question of, okay, what do we do? So we're gonna go to a field. We're gonna go to a field and we are going to plant 6 cultivars, 3 with Peking source of resistance and 3 with PI 88788. We're gonna treat it with a seed treatment that says that it can manage soybean cyst nematode. So Ilivo will be the one that we're gonna use. Fluopyrin is the active ingredient. The field is hot. I mean, 13,000 to 15,000 X per 100 cc of soil. I show you results with 5,000 reducing 37%, right? This is massive. Is this true? Yes, because I work with a grower that has 27,000 X per 100 cc of soil in a composite sample. The population is one that can reproduce on both 88788 and Peking. We're gonna get the reproduction factor by dividing the final population divided by initial. If it's the same, it's 1. If it's more, it's gonna be above 1. If it's less than 1, and that's what we want. Yields in kilograms per hectare. Sorry, I didn't put measures per acre this time. And this is work that we're doing with Vinny Garnica, a postdoc in my lab that just now recently got hired by Washington State University. So very good. It's gonna be a colleague of us now. This is our field. Of course, this will call, if you think about what you have here, I know that you're thinking split plot. You need to do a split plot and you need to put it, arrange it in a block design, right? Because you wanna have your cultivars as a whole plot, with and without treatment, your small plot, and then you wanna have your plots. Because you're doing a field experiment, you want to account for variability. That's what we learned, and it works. I'm not saying we don't have to do that, but at the end of this talk, maybe I will. But this is a problem, right? This is the challenge that we will run into. That's two.
You said.
So the thing is that when I go and walk those plots, those blocks, by definition, blocks will have all the treatment once. And when I go and I walk those whole plots, and then I walk those subplots, and I sample them, I see that there's a 6-fold difference next to each other. So, I did everything I had to do to have a control experimental design in the field, yet I have this spatial aggregation that will translate in my response variable, and I will severely violate my ANOVA assumptions. Spatial aggregation violates ANOVA. These fields, like I mentioned, you will see high variation in the numbers. Blocking alone is insufficient for what I just explained. 2 years, spatial model, 6 spatial model tested, and 176 field plots. This is the first time that I started getting into the habit of putting all my data in a repository and sharing it with everyone. So if you would like to go and download this data and run this analysis or a different type of model, and then say, hey, dude, this is actually better, welcome. This is something that I'm trying to do now in my lab, try to share all this data whenever possible. Sometimes we can't, but when we can, I will. 6 models that we will evaluate, okay? So we're gonna start with a baseline. That's our split plot, a randomized complete plot design. We are going to then adjust for the SCN. So we're gonna use SCN from each plot as a covariate. Works very well biologically speaking, right? But still doesn't capture a lot of the spatial variability. We're gonna use AR, the autoregression model that says, hey, I'm gonna assume that the spatial aggregation is the same continuously. That may work, but maybe doesn't capture large scales. We're gonna add a nugget to that model, which means that, okay, you're gonna have some spatial variation next to each plot, but also we're gonna put another coefficient that will account for some random noise. Then we're gonna do a trend adjusted that will include rows and columns and will capture that spatial variability at a large scale. And then finally, this is our winner, the tensor product penalized splines, which will include local and large scale accountability for that spatial aggregation. So we will account for that and uses a non-parametric approach, so then it doesn't impose so many constrictions to the model. Yeah? So we have 6 models. In theory, we're trying to account for more and more variability in space. And the only thing we needed was the row column position that we normally have in our map. You go to a field, state 101. That's it. That's your 101. That's your row column, and so forth and so forth. And then we'll keep the split plot. We'll still keep the split plot randomized block design. So how well does it work? We do multiple comparisons. We look at the residual AIC, BIC. But one of the things is when we don't account for spatial variation, this is the nematode. You see that it's very rough, the raw data after using TPC, right? We see that everything smooths out. And it's smoothing out not because we're changing the data, it's because we're accounting for the spatial aggregation. And same thing with the response variable. Very core hotspots that then gets diffused because we're accounting for the row column spatial variation in the data set. And then we use relative efficient, which is the comparison of how precise our models become compared to the base, which is our split plot randomized block design. So you see that there's an improvement, 10% better in precision of what I'm going to inference about. Essentially, that's what it's telling us. As we go to a more sophisticated spatial modeling, we see that we improve 30-something% compared to the baseline. And what does that mean? That means that without adding reps, extra plots, you are increasing that in your analysis. Yeah, so it gets a better compared to the baseline. That's the so and we use other stuff, but this is showing us that it's working. So then we go with TPS, and then we can conclude saying, hey, picking over PI 8878 produce on average more for that population, and it reduces 36% the reproduction factor. This is great because it's giving us a little bit more yield, a little bit, because if you do this on bushels, it turns to be like 3 or 4. But this is a big win. It reduces the reproduction of the nematode. The protection gives us a bump in yield. However, it does not reduce the population of the nematode. So it's a good yield protectant. There are other stuff. There is fluopyran. It's a good fungicide. There may be other stuff that is taken care of. Bump in yield, no reduction in nematode. And then the gain in efficiency by using these models is a major win here. A trade-off between suppression and yield stability. So you see that at these low levels of nematodes, Peking is producing a lot, and on average, that's what it shows. But as we have more nematode, unfortunately, Peking is losing more yield. So as we have more nematode that can reproduce on Peking at a different level compared to PI 8878, PI 8878 seems to have a better buffer in terms of protecting that yield loss. And Peking just goes down very quickly, which is one of the reasons why we plead our growers not to plant Peking continuously. They're gonna do it the first year, they're gonna see, I reduced the nematode population, my yield increased, I'm gonna do it again. And then you're just gonna allow that development of virulence on that source. Good. Diversify resistant, again, don't stick with one. Fluopyran strategically, you're consider seed treatment. Again, this is something that costs money to the growers. Monitor SEM virulence, getting that female index above all, just not staying with this AG type or race or whatever you want to call it. Use how much is actually reproducing on that. And adopting spatial modeling really doesn't take much, but geographically telling us where those plots are, row columns, and you already have it. You have it in your map. And it can be very easy to be part of an analysis. So we did that in small plots. Now I want to take it to an on-farm trials. I want to show growers what we're seeing and how their populations are probably damaging their soybean and what we can do to reduce population. So Richard Gonzalez was a master student in my lab, and he used some geospatial statistics or approach to go and test an automatic soil sampler. This automatic soil sampler can do the job of 8 people, 9 hours in the field in 2 hours with 1 person. Amazing. How does it work? He has a knife. He throws that knife in the floor 6 to 8 inches deep, pulls it out, and then he has a blower. That blower will collect the soil sample here and also will clean that knife very well. That soil sample then will go and will deposit on these little plastic containers. Those plastic containers will have a QR code linked to the GPS that the tractor has. So every time that knife goes down, we get an XY. And now we can get the amount of nematodes for that XY. Pretty cool, huh? And now Richard can say, well, I'm going to go and work with growers, large areas. We can test Peking and PI 88, 1088, El Livo. And we're going to first have to test if the automatic approach really concord with our results in the manual way of doing things. So here we go, manual and APSS, automatic precision soil sampler. The black dots are the places where we sample manually, and then when the machine went and sampled, the maps are pretty similar, right? The maps are pretty similar, and then you can do a interpolation using maybe inverse distance weighting and creating these raster maps files that now you can look and identify hotspots. You can go now and place your trials where you actually have nematodes. You can go and talk to a grower and say, well, I understand that you have soybean cyst nematode, but you don't have too much soybean cyst nematode here. Do soybean here and do corn here. You have alternatives if the market shows like, more price for one than the other. Soybean, of course, is the best crop, so this is soybean. But we are seeing the same thing at a large scale. The same thing that we're looking and observing with our small plots, the same thing we're seeing with the grower. And they are seeing it. They're seeing it that their PI 88 is tremendously losing yield. Peking is holding it better for that population, but still there's a little bit of slope there, not significant. And my seed treatment didn't do much in my field. So the SEM coalition, again, I showed you this before. Rotate, rotate, and rotate. Rotate to a non-host, rotate sources of resistance, rotate varieties. And then if we want to put this in a 6-year plan, let's say, plan something that is easy to find out there. Non-host, go with Peking. Non-host, avoid Peking. Try to plan something different, maybe. Good. How are we doing? 4 minutes. Good. Any questions? Burning questions? So I'm going to jump now into talking about the SEM coalition and what I'm doing there, my role, and why I think it's important. We recently wrote a manuscript talking about this. I was invited to give a talk in the SOM meeting in Canada. I don't know if you were there, they saying. But a manuscript came out of that. So hopefully this piece will be available soon. But, adaptation is one of the things that we're very concerned by all the evidence that I show you right now. And the SEM coalition has many farmers. And what we're trying to do is communicate about SEM and management with one voice. So it's a public-private partnership. And the reason that I'm saying one voice is because we want the consultant, when they talk to growers, to communicate in the same way that the private partner, the private company is doing it. The extension educator is doing it. The state specialist is doing it. And you see the nice group of collaborators we have. Raise awareness of SEM resistant problem and encourage more growers to actively manage SEM. That will be the goal. This is what it looks like. A lot of videos, clips. Couldn't find yours. I was looking for your video. I don't know where it was. You didn't? That's why I didn't find it. Power of partnership, we go and we're invited to give talks to some of the workshops or webinars that industry will do. We go as the unbiased source and SEM Biology 101 type of speaker. So Greg Tilga is talking in a Syngenta event. I was invited to talk in a BASF where they released the name of a new product that is coming in 2028, Nemasphere. It's the first GMO that can protect soybean cyst nematode. The lifetime of the coalition is captured in many numbers. We had a front page of the Progressive Farmer. This is a, this is like a GQ or like, I don't know what example can I use. This is a really, a cool magazine. Then we got front page, and then there was several stories within about SEN and research. And it doesn't have to be SEN Coalition. We invite collaborators to write about what they're finding, right? But you can see the use that our website have. The SEN Profit Checker has more than 82,000 users in 2025 January. And again, it's something that is moving us away from those tables that are so confusing and allowing us to use data-driven approach to know how much soybean cyst nematode can reduce yield in that field. I talk about this just now, but this really translates into us going and talking to growers in front a field that is highly infested with soybean cyst nematode, yet beautiful. How beautiful that soybean field is. That's loaded with soybean cyst nematode. And having growers there, listening and going and pulling that plant and seeing how loaded with the nematode is, it just makes a difference when you try to convey a message. We also talk to growers a lot. We produce a lot of materials that we get their input. I mean, what do you want to see? Life cycle, disease cycle? No disease cycle. So we listen to them. We go and do our life cycle too, but we listen to them and we put materials there that are more of the things they want to use, and they will be practical for them. We go and we use research surveys to try and capture what is it that the SEN Coalition is doing. So we partner with an independent firm that can do this. So that's the third one. So now I need to stop easing, right?
You can go another.
No, that's what I'm saying. So now I'm gonna start, relaxing a little bit. So I'm gonna start doing here. So, really cool data-driven, impact information that we can gather. We send a company and they will create a bunch of focus group with growers or send surveys, and then they will process that at different beast, right, in terms of statistical analysis, processing survey data. But it shows that since 2015, with the message of the SEN Coalition, farmers soil sampling for SEN have increased significantly, right? And then we want to know also, well, are you switching your resistance? Are you considering resistance where you're planting? And clearly since 2015 until 2024, it did, right? Same thing for Peking, which is now available and more available to growers. Before it wasn't that much. Clearly now that they have availability, they're planting that. And then the last one was seed protectant. It also allows us to identify gaps, these type of surveys. So the growers, when you ask them, do you believe in soybean cyst nematode? Yes, I do. Do you know that it can cause yield reduction? Yes, it does. How much? Well, 5 bushels per acre, a lot. Well, our data shows that it actually costs significantly more than that. So what's going on here and how can we capture that really? So this type of research, the survey research, allows us to do that. And this is what the SEN Coalition kind of look like in terms of people that are running and working and doing. We're putting together this. Me and Dylan Manjul from University of Nebraska, we work a lot with Morgan Meyer. This is our communication firm. So we do all the reading and try to make sure that the science is there. But we work with an advisory board and the previous SEN Coalition leaders. So whenever we're in doubt, we know that we have people that have been in the field for longer and can help. And we meet regularly to update on the things we're doing, brainstorm, and then invite our peers to be a part of this, as you see in many of the videos. The expansion of the SEN Coalition, SEN stands for Soybean Cyst Nematode, is, well, now we want to move to maybe aborting other nematodes that can affect soybean. Because in the southern part of the United States, soybean cyst nematode is not a big deal. Root knot nematode is a major constraint for yield, or reniform is when you have cotton, or lesion nematode, Pratylenchus, across the US. So last year, or this year actually, we put out 4 management guides. We put it on an interactive website, so you can go to the SCN Coalition and hit the field guides, and you will be able to navigate there from your phone, something that the grower told us that they want. They just want to do it from their phone. You can download those guides as well. We updated the SCN management guides that was outdated for a decade, so we updated that. You can see that we have many co-authors and many reviewers that help us make it better. We created 3 new ones: the root and nematode management guides, the reniform management guides, and the root lesion management guide. And again, you saw Darcy there helping with that, Lee-san help with. So thank you for that, for your contribution. But this is now available to growers across the US. Well, I'm in Arkansas and I don't deal with SCN, I deal with RKN. Now you have a management guide led by Travis Foskey, expert on RKN, and other co-authors that you can download. Well, I actually deal with reniform because I do a lot of cotton. Well, the one that Kathy Lawrence from Auburn led with other co-authors, you can download that. Well, what about lesion? We have so little information about lesion. Ann McGowan, University of Wisconsin, leading with other co-authors, put the management guide for lesion. So expanding from SCN to other plant parasitic nematodes that affect soybean. And with that comes new infographics. We're dealing with other animals, migratory endoparasites, which is different than a sedentary endoparasite. The sampling should be different. So we account for all that in our different management guides. Okay, and with that, if I need to give you one message, which is broken in several ones, right, will be that the SCN is an evolving pathogen. Every time I say this, so one of my favorite books is Alice in Wonderland, okay? And then you have Alice in Wonderland and Alice through the looking glass. And through the looking glass is where the Red Queen grabs Alice, right? And say, hey, now let's run as fast as we can to remain in the same place. And if we want to get somewhere, we need to run twice as fast. That's what I feel when we work with this pathogen. We need to run as fast as we can just to stay in the same problem, and twice as much if we want to make a progress. Virulence is quantitative. Try to move away from those binary or categories. Female index is that, and then spatial structure. We know that soil-borne pathogens are spatially structured. Why not account for it? Integrated management is essential. So resistant, seed protectant, and sampling. Spatial modeling, improve decision making, more precision, better inference, no extra field costs really. If you have, I know you have your row columns, I know. We all have maps, planting maps, and those have row columns. And then the impact that we're generating when we're working together and thinking about the message that we send to the grower. The grower is too busy. They don't need to hear one story here and another very different here. One a message. And with that, I thank you very much. I want to also thank my team and all the farm managers that without them, I would not be able to be here showing you all the data that I have. Thank you.
We have time for questions.
So you had 7 or 8 sources of resistance, right?
Yes.
Is it that those are from wild species and it's hard to breed them in, or why?
That's a good question. So no, they're all Glycine, Max. They are wild in the sense that the black coat, black seed, the species is the same, but they're still Glycine, Max. They will never grow in a field. They will grow viney. They will grow. As a matter of fact, in February, I grow a bunch of these and I give it to my wife for Valentine's Day. And she doesn't appreciate, but they have beautiful flowers, purple flower. So yes, and maybe you have a follow-up question.
Is the resistance linked then to that?
Yes, that's a great question. So the question is that resistance linked? And there is a paper that came from Terry Niblack lab where they show correlation of how these resistances are overcome. So 7 is always overcome first, 5, the one that is on number 5, and then 2, PI 8878. So whenever you have a 257 and you look at the female index is higher in 7, then in 5, then in 2. And same for the other one. The only joker there that it's like a miss card or a wild card is at number 4 that we talk about. Yes, and they try to incorporate that in high yield in soybean, commercial soybean, and didn't work. Now we also, you didn't ask this, but I'm gonna tell you, we also identify populations that were able to reproduce on that number 4 very slowly. We couldn't have enough to do research. So then we say, well, now that it's growing on number 4, let's put it on a susceptible, and then let's put it back on number 4. When we put it in the susceptible and then back on number 4, it didn't reproduce anymore. Super weird. And I didn't reproduce that, so this is anecdotal, what I'm telling you.
Okay, yes. So it seems like the field sampling has been solved. That problem is solved, it's all automated. How about the lab portion?
No.
So I know,
Yeah,
I think I've seen something about
That's a good question, man.
imaging, but what else has been done in the past 5 years?
Yeah, that's a good question. The answer is no. Still, Richard had to process all those samples. We had to calibrate depending on what field we went to sample. You need to do a bunch of your track of that automated sampler going because it doesn't capture 100 cc of soil that you need. And then for the imaging, I know that Greg Tilka has a really good setup of automating the counting. Still, they have to process. And he showed me this when I was there in Iowa State. I went and visited his lab, and he showed me the software is very accurate. And then I said, well, I want this software. And the company that made that software is not around anymore. So he has a last prototype there in the lab. But we have been playing with some of the new softwares and that came out. Like there's an R software, there's a Python one that takes pictures, but you need again to take those pictures and feed it into the program. Yeah. I know that people are working, but that will be the next place to alleviate the delay on processing some. Yes, sir.
In your map of Ohio, you had everything, it seemed like all of your SCN was localized up near the lake, right? So is that soil texture, topography?
That's a
Is
great
it
question.
Cleveland's?
It's Jana.
Yeah, so this is where Ohio grows a lot of soybean, the northwest part of Ohio. Okay. This is it, right?
Yeah.
This is where I love to go and give talks. Because this is the beginning of the Appalachian Mountain. So it's super hilly and really very few soybeans that are doing just because, we're gonna go and do it and we're gonna get some money out of that. Very few farms there, this region here. Then again, here in this northeast, close to Pennsylvania, again, you start seeing more farms. But that's really the reason when you go here, Hawking Hills and all those places are that in nature. And then here you have the heavy soil, so really good production here and a lot of Phytophthora too. And here in this region here, a little bit more sandy. Here in this region, a little bit more sandy. You see more of that damage of the nematode there. Then the other problem that we struggle here in this area, growers that have been planting soybean for 30 years. And they will not plant corn. No matter how much data I show them, they will just continue doing soybean. Yeah.
So those gray counties have never confirmed.
These gray counting, no, they're never confirmed. Sometimes we don't even receive sampling from them. So Hawking, we rarely receive any sampling from them. We're always asking. Sometimes when we're driving, we go and we just go on a field and get it.
Yeah, I know. Have you seen the Indiana map? See our.
Yes.
So we have the one county.
Yeah.
There's no beans in that county.
No bean on that? So that makes sense.
Or little to no that we have.
That makes sense. Now,
I always get half harassed on
the
that one.
soybean seeds nematode can reproduce on other hosts too. Soybean is the most common agronomical host, but it can reproduce on some of our annual wheats. So purple dead nettle, for example, now you see beautiful fields that are purple. Those can allow the nematode to reproduce. Pennycrest, there's a push for pennycrest that will allow the nematode to reproduce. There's handbeat is another one. Hemp is another one. And then some of our cover crops, some of the clovers, for example, will also allow them to reproduce. Yes.
It was a really entertaining presentation. So thank you for that. And I have a question, and it's not regarding about SCN, but it's about red crown rot. So I know that there is one county, and you sent us some data, some sample from that. And I was curious because it is like in the center. It was Delaware, right?
Yeah,
So
Delaware.
it was like it was in the center, right?
Yeah.
Have you ever surveyed like for red crown rot like in the other states?
Yes, that's a good question. So red crown rot is a different pathogen, it's a fungal pathogen, and if you have interacted with the Talenco lab, it's all over here next to Ohio. So I took my chair and my coffee and I was there. I sat down in the border of Indiana just waiting for red crown rot. And all of a sudden it popped out in the middle of Ohio. And the reason we think that is because, they're really good at identifying SDS. And I have growers that say, I had a little bit of SDS, but I don't worry about it. Well, that could have been the beginning of red crown rot in your field, right? So we did identify there, and then we went and we went around the counties, but there was really not symptoms, not even of SDS that year. And I believe it started very warm. That was not, and started warm, and it rained. So then growers planted later. They didn't see SDS. It was great for red crown rot because it's warmer soil temperature. Maybe that's how we see it in this field. But also this person planted soybean for more than 10 years. Okay, see you. Any other questions? These are great questions, by the way. Yeah.
One last question, maybe. So for the HG tabs, right? We successful check, different lab, they may use different variety. So do you think like you use the Lee 74?
Oh, that's a
some
good
use
question, man.
Williams 82.
Yeah.
When you compare from different starts,
Yeah.
doesn't matter.
Yeah. So you're saying why not use other susceptibles
Yeah.
as a check? Is that the question?
No, I mean like you use the Lee 74, right? Like
Yes.
for example, we use the Williams 82.
Yes.
But if you want to compare the film index from different labs, I mean patterns of finding things, does it matters?
So no. So in 2009, there's a paper in Plant Health Progress that is a standardized greenhouse assay, 2009. We compare different susceptible check and see if they change the female index that will be significantly enough to change your call for AG type. And we didn't. Hutchinson, Williams 32, Essex, Lee 74. All of those you can use. And that's a great point because Lee 74, it's so picky. You grab it and it like just wants to run away. It doesn't want to roll. It doesn't want to germinate sometimes. So we did that comparison. The other thing that I did, because I'm always freaking out about like, am I calling this right? I sent my sample after phenotyping to a lab in Missouri, SCN Diagnostics, a bunch of them. They did I pay for that. They did, the testing, and then I compare my calls, and they agree. Now, the careful thing to have when you do this type of test is that you have reps. And one of our rules in our protocol is that you grab those rep and you find your coefficient of variation. And if it's above 30, for our case at least, we redo that. We discard that and then we redo it again. So Gili had to run a bunch that he called redo because his coefficient of variation was large. And we don't feel confident calling something AG type 1 or calling something resistant or moderately resistant when we're comparing genotypes from a breeding program. Over there in the back, any? You?
The Peking seems to be packed with resistance genes. Right? Peking, the
Yeah.
the source of resistance, right? How many genes does it have?
At least four.
For resistance.
Yeah, so RAG1A, which PI8878 has RAG1B. And since 2012, we learned that it's not only the fact that they have the RAG1B, but it's how much of that copy numbers of that
Yes.
they have, right? And then you have RAG4 for Peking and other minor genes. And now breeders and nematologists like, Skabu and Michum are understanding better and know that more pieces are involved in the.
So why do they need to cross it many times to bring a variety to? Why is Peking preferred? Not only because of the resistance genes, or because they are linked, maybe it's easier.
So the thing is that we have imposed a selection with PI88 and 88 across North America to now have the most prevalent population able to reproduce on PI88 and 88, which in most cases cannot reproduce very well on Peking. Peking didn't make it to the market available in time until recently. So for the past 30 years, we have been just hammering the same population with 88.788. Peking made its way in, but it was really effective at protecting the plant from the nematode, but it also unfortunately brought a lot of issues and the yield wasn't good. So it got stigmatized a little bit. Now you have really good cultivars that yield really well that has Peking. Now the problem that we are seeing with our analysis is that even though we messed up PI88-788, it still protects the soybeans. And we're seeing that Peking more quickly will be able to be adapted by the nematode. Or I don't want to use breakdown resistance. I don't like to use that, but in a consecutive soybean year that you do Peking, you may see adaptation of that population on Peking. It works really well, but those who escape that will reproduce very fast on Peking. And then you lose both sources of resistance.
So do you know what the GMO is, what the gene or the mechanism is? The new 2028.
Yes, the new mechanism is a cry protein. So it is essentially a BT. If you're in corn or you learn about Bacillus thuringiensis BT GMO, right? So it's a cry protein. It's not exactly the same. I think it's cry 16 or something. It's published out there. But the way that it works is the nematode will ingest nutrients and those cry for crystal will go and disrupt the intestine and kill the nematode.
Yeah.
Oh. Thank you guys.